name is Sergio Rocha. I'm original from Portugal, but I live in Norway for the last 15 years. And I'm working at an MBU for the last 10. My background is in marine biology and fish nutrition. And I'm working with Professor Margaret Overland for the last 5. Um, so today I will today I will talk mostly about the feed technology and the microbiota. That was the two things that I learned or uh, specialize after the nutrition part, which I think that are extremely connected and relevant, and to take that both into consideration. I will proceed to my presentation. Let's see if I can make it happening. I guess I have here a problem with my screen. You are not seeing the presentation yet, right?
No, we are seeing your PowerPoint.
Yeah, I will make it happen. Better. Yeah, I think that now you will be able to see it.
Yep, there we go.
I just need to fix, that is the part of the presentation is also, yeah, now everything is correct.
Yeah, we are good.
Yes, so the first thing that I want to talk, or the way that I would like to talk to start is like I said, that the nutrition itself, it's a part of a whole where we have the immune system, the microbiome, and the metabolism in tight connection. And it's not nutrition itself, but also the microbiota and the connection between the microbiota and the immune system that are together will make or promote a good fish health. And again, they are really well connected in their close relationship. So much so that have been proven or demonstrated that when fish are in the germ-free conditions, there is a lot of intestinal epithelial cell dysfunction. There is also some impaired nutrient absorption, more vulnerability to pathogens, change of metabolism, immunosuppression, reduced growth, and higher mortality. So they've proposed that microbiota, it's actually should be considered an annex organ because fish totally depend on it. And also, before I go further, I also would like to do a disclaimer, the difference of microbiome and microbiota. There is a lot of people that thinks that is the same, but they are not. They are pretty similar, but not exactly the same. So the microbiome is the collection of the genomes from all the organisms in the environment, while microbiota are the microorganisms themselves. That can be bacteria, virus, fungi, so on. So the microbiota in fish, as in the other animals, but today we are focused on the fish, they work like a society, different roles, different importance at different time points and different locations. And they can have some important functions related, for example, with energy storage or growth. They can, for example, have regulate the appetite, or they can produce some enzymes or vitamins. They can also interfere on the density and size of the villi, so indirectly they affect the nutrient acquisition. As I mentioned, the appetite or the intestinal motility, or can also influence by the production of short-chain fatty acids, or that, for example, have butyrate, that is the main source of the epithelial cells, or they produce some lactic acid, or some antimicrobial peptides. But the antimicrobial peptides, they actually are more related for the immune system. or the interaction with the host in the mechanisms of defense, because these microbiota, they can activate or support maturation of the immune system. They can also the defense against pathogens, like I said, and also the competition for space and nutrients. So in the way of if an, a bacterial composition is well balanced, it means that when a pathogen comes, doesn't have room to grow because there is no room, there is no nutrients. But if the microbiome, the environment is not so balanced, then the pathogens, they have a space to grow and prosper. And also the microbiota is also related to promote the health, to promote the fish development and the lifespan. So in other words, less mortality. When we go more in detail about these soft energy storage, I can just give some examples. You have the core microbiota are the ones that most of the time are always there, and all the fish from the same species they share. Or we have some other bacteria with special functions, like lactobacillus or bifidobacterium or bacillus, that they are related, for example, with the production of lactic acid. All of these small examples, they were already mentioned the slide before, but I just, with some more examples of short-chain fatty acids or enzymes or vitamins, that which the host depends on it. So this is the host benefits a lot of these these metabolites, while the bacteria benefits of having a place where they can grow. Related with the immune response, there is a lot of new data these last years to prove the interaction between the immune system and the microbiota. And as I mentioned here, the colonization resistance, the first come, first served. So when the space and nutrients are well used and distributed, there is less chance of the pathogens to colonize. And some examples of antimicrobial agents. I have here in the corner some bacteriocins, organic acids, hydrogen peroxide. They have been demonstrated that they are a well-developed mechanism as a defensive mechanism, which are regulated by the microbiota. I always like to point out that when fish are stressed, it increases susceptibility of pathogen colonization. It is something that have been demonstrated in the last years, and is something that we should also. The stress is not systemic, but can also be in the intestine of the microbiota. So if the microbiota is so important, how can we regulate it? Well, there is a lot of different complex systems, as you can see, that will end up in the red on the gut microbial community. But I don't go through all of this. This is way too overcomplicated. So we'll keep it a little bit more simple. So there is some biotic factors and abiotic. The biotic factors, they are related with the development stage, the immune system, genetic, or the sex of the fish. And the abiotic, they are more related with water quality, the water characteristics, or the diets, where do they live, or the stock density. Today, we'll focus a lot on the diet, but I will mention a little bit more the others. So the fish, to start with, fish can be fed with the prebiotics or probiotics. I mean, normally I just oversimplifying, saying that the prebiotics are non-digestible ingredients that selectively stimulate the growth of intestinal microbiota. In other words, it's food for the good bacteria, in quote marks. For example, fiber. On the another one, the probiotics, they are live microorganisms that can colonize the intestine. When I say can, is because it doesn't mean that all the probiotics that you ingest or the fish ingest, they will go to the intestine and colonize, and you don't need to ingest them anymore. Actually, it might be that the fish requires a probiotic to be continuously provided, because if you are working with a relatively healthy fish with a well-balanced microbiota, they will most likely not be able to colonize. So this is something that you always need to pay attention, that they don't need to be colonized in order to have a beneficial function. But then there is other strategies. So besides the prebiotics and probiotics, as I said, you can go with some symbiotics strategies. Is when you introduce one prebiotic and one probiotic, or a combination of those. So you are actually, for example, providing live bacteria together with additional fiber, and they will most likely have a more efficient role. Or you can have paraprobiotics or postbiotics. Paraprobiotics, they are also known as the ghost probiotics because they are dead bacteria, inactivated. Or the postbiotics is when we actually provide the metabolites produced by the bacteria, or if you have a lysed bacteria that will release the metabolites inside of them. So those are considered postbiotics. There is a lot of papers that already have been documenting the importance of that for aquaculture. So it's not such a new things. And now besides diet, I want to go through other other factors that change the microbiota. For example, the age and development, because fish, in theory, when they hatch, they are germ-free, in theory, and the microbiota is acquired from the surrounding environment. So through the time, from the fish, from the egg to the larvae, they will start develop a more specific microbiota, which could be related with hormones or also related to their first feeding. So it's related. Have been, as we said in the very beginning, it's have been shown that they have a better development when they have a good microbiota, or at least a more balance, or what it expects to be related with their functions. Also, if fish are used antibiotics in the very beginning, they would most likely have um, some problems in the later stage related with locomotion or development. Another factors that we have is the genetics or sex. For example, when they have carps in the same pond with the same microbial communities, but they have different abundances. That was related with the species of the carp. Or in other examples, when they have the same species, but females and males, they could have different microbiota, and most likely it was related to hormones, but it's not observed in all the species. Another case that regulates the microbiota, it's the trophic level of the host. So when you compare herbivorous with the carnivores, they most likely would, or most likely they have completely different microbiota because they have different requirements. So the herbivores, they need something that are more bacteria that are related with the fermentation, while carnivores and omnivores, they need more bacteria that aid to the protein digestion. Because they need to produce more enzymes related with, for example, proteases and chitinases, that they are important for the digestion of the fish. Another factor, it's where do they grow. If they grow in the wild, they might have richer and more diverse microbiota, and with several functions. As you can see here, some examples with Schillen degrader, sulfate oxidizer, and sulfate reducer. While in farmed fish, they are more related with nitrogen fixation and denitrification groups, because they have all the nutrients that they need, the feed is processed. So the microbiota that they have, it corresponds of the environment that they need, not just the diet. And then, for example, the salinity, obviously, there is some bacteria that tolerates different osmotic pressures. So when fish go to salt water, they have a different bacteria than the ones that, well, they were in seawater. It happens in salmon that when it goes from one environment to the other, change the diversity, but also the bacterial load. And the temperature as well. Uh, so um, bacteria have different optimal temperatures to grow. And for example, if uh, bacteria increases, theoretically the bacteria will grow as well more, but it can grow in the way that is not favorable to the fish. So, for example, when the bacteria goes up to a certain level, some important bacteria can be um, decreased. In these cases, with some lactic acid bacteria, and the fact that they stop being present, or they disappear, or they die, then it has space for pathogens to grow. In this case, the Vibrio. So higher temperatures, besides stressing the fish, change the metabolism and so on, it will also automatically change the microbiota and makes the fish more vulnerable to pathogens. Obviously, another effect is the use of antibiotics. So when you use several antibiotics, you um, decrease the microbiota load. So it increases the vulnerability of opportunistic pathogens, the same as the temperature. And another fact that we need to concern about after all of this modulation, these conditions, abiotics and biotics that change the microbiota, is the dysbiosis. I always give a little pause here or mention, because we need to think about what is dysbiosis. A lot of studies, they just say that is a unbalanced microbiota. Like I have it here. This is the opposite. This is a eubiotic, as it's well balanced. But dysbiosis is not just unbalanced microbiota. The dysbiosis has actually more than one thing. It's the term that is used and still under scrutiny, is that the stability of microbiota community and the ability to recover and return to the balanced state. However, this unbalance when unbalanced, it needs to be associated with a disease or susceptibility to a pathogen invasion. So again, so it's not just because some people present results saying, oh, the fish has dysbiosis because they have an unbalanced microbiota. That is not enough, because it needs to be associated with the disease. And also it's if the fish is able to return to the balanced state, then it's not a dysbiosis. It could be something that they could be some temporary diarrhea, for example, or something that toxin that come through the algae bloom that is in the short period of time. So if something change a lot the microbiota, but if they are able to go back to the balanced state, that should not be considered dysbiosis. Yes, and a way that we could measure dysbiosis is proposed by this paper by Mogin et al. That they say if there is some bacteria that goes up or some bacteria that goes down, I thought that was an example here. If some of them, for example, if the tenacibaculu go up, no, if Mycoplasma go down and the Alivibrio go up, uh, normally is associated with tenacibaculum, so it might indicate it's most likely considered a dysbiosis. Or on the other way, if in the skin, if we have some protobacteria that decrease and the flowbacteria that go up, and that normally could be associated with viral infection. So what I mean is that you need to have an imbalance associated with a disease before you state dysbiosis. So here was just a small break for the next topic. That they need fiber. Bacteria needs fiber. They need food. It's not just who is there, but we need to keep them in good health to help us or help the fish. So as a main conclusion so far about the role of the microbiota is that the fish intestine harbors hundreds of species in the complex equilibrium. Fish depends on the microbiota, should be considered as an X organ, and genetic and diet are some of the major drivers for microbiota modulation. Microbiota is responsible for key physiological functions, general health and growth, and is also involved in the immune response and defense mechanisms against pathogens. So, manipulating fish microbiota has a great potential for aquaculture to improve animal welfare and growth. Then we already know, so hopefully we all agree that microbiota is important, but the next question is how can we actually measure it? Or what are the steps, which are the considerations? So in the general approach, we collect digesta from the fish. We collect the total DNA. We amplify the 16S gene. We sequence, and then we analyze the data. I'll go briefly in a little bit of all of these steps. Yeah, and if there is other further more detail, then we can talk in another moment because there is a lot of things that can be talked about it. So normally I use the column method that is a kit that is for isolation of DNA from stool for pathogen detection. We don't look for the pathogens, but you're looking for the bacteria. And from all different kits that we use, this was the one that works the best for us. So it's a column method. We just collect the digesta. We have some digestion of proteins. We have some binding to the column and washing, and it's a really simple protocol. However, we need to do some readjustments because this was made for humans, and the salmon, in our case, has much less bacteria than human feces, and there is much more DNA from the host, which we don't want to. So we need to change some temperatures and some lysis, buffers, and so on. But everything is described in the papers that we publish. But the point is, if you work with a new species, perhaps you need to do some testings before you just go processing all the samples at the same time. Besides the digester, I would like to point out one thing here. Even though here I just say that we collect the digester sample, it's really important to, if possible, if working with a big fish, to collect also mucus. The reason is we have two different communities in the intestine. We have the ones that live in the mucus, that are the ones that are more related with functions that the host is really dependent on. And then we have the bacteria that are transit, the ones that are going through the digester, that they are not necessarily colonized. So those ones might have more a less significant function in the long term, might be a short-term function, when the ones that are in the mucus are long-term. But if you work with small fish, it's really difficult to collect mucus enough to collect DNA from. You can collect mucus, but might not have enough DNA from bacteria to process in the normal standards conditions. So it's really nice to, if possible, to work with big fish to go for digesta and also the mucus, something that we try to do every time that have big fish. So as I was saying, besides that mucus and the digesta, we also have some feed. We collect some we check the bacteria from the feed, and the reason is if something happens in your results, if you have a pathogen, you kind of want to know if it was a pathogen that was present in the feed, because the 16S sequencing only analyze DNA. They don't know if the bacteria is alive or dead. It could be some bacteria that came from some ingredients during the feed production that was carrying along but doesn't have any biological function because they are inactive. So it's really important to also to check in the feed. Something could come from the feed. We also use water that for the same reasons. We want to see if something weird happened in the digester. We want to know where it came from. Could be just environment. And this mock, I will elaborate in the next slide. I will explain soon. This filter paper is because we need to filtrate the water. So we want to see as a blank. We want to see if something comes on the water. We want to see is from the water or from the filter paper. And lastly, a blank, that is a control, negative control. This mock that I'm talking about, what it is, it's a specific community that we know which bacteria is there, if they are gram-positive or gram-negative, and we know their relative abundance. So what we do is to use it as if it was a normal sample. We do the DNA isolation, we do the amplification, all the steps. And if it comes pretty similar of the theoretical, then we can guarantee that our extraction and our sequencing, they were correct. That is really important to add and gives an extra layer of quality of the data interpretation. The water, I already mentioned, we have filtrated the water, and then we have these membranes that we use also to the sequencing of the membranes, because there is a lot of things that comes from the membrane because it's a biological product, and that is not in the water itself. Let's say, how does this amplification works, and how can we later on, how can we interpret those data? So we follow just a protocol that is pretty standard from Illumina. And for the ones that don't know, what is this 16S? Why do we go for 16S and not other gene? The reason is that the 16S is a ribosomal gene that is present in all the bacteria. And you see that that is in this ribosomal DNA, RNA, they have conserved regions in green and the variable regions. So the conserved regions are the same in all the bacteria, and the variable regions are the ones that we use to identify which bacteria are there, which bacteria it is. So if you see more in detail, there is the one that we use are the V3, V4 in our case. It means that we have primers in the we sequence the variable zone 3 and the variable zone 4. We have primers in the two conservative areas. And because we use the MySeq Illumina. But if you want to go down to the species level and know really all the details as possible, it's also possible to do a full length, but those are much more expensive and time consuming and more technical. But it's possible to, depends which application, which species, which protocol you follow, there is different areas to sequence. So it's not just black and white. The sequence, as I said, we do through the Illumina MySeq. Again, it's nowadays there is a lot of companies that they can do it for you. It's not every lab that have a sequencing machine. It's pretty expensive, but with more experience, it gets easier and cheaper. So now I will just give some course or really small introduction, or how can we analyze this microbiota, because it's really, we already know that they are important, but a lot of times we come these plots and we don't know how to interpret them. So this is a simple example of freshwater and seawater from the same experiment. And just by looking at them, this is normally relative abundance. We also need to pay attention that they are not amounts, they are relative abundance. And the first, they are at phylum level, which don't say so much. At phylum level, we normally want to go to lower taxonomic level groups. And then also, it's really common that we see that even though in this one we see the difference in the abundances, then comes these diversities, alpha and beta diversity, that a lot of people don't know what they mean, and I will try to debunk that today as well. So for the alpha diversity is a metric that values the individual intestinal microbiota profiles detected in this Atlantic salmon, which the beta diversity results of intestinal microbiota profiles detected in Atlantic salmon. Doesn't mean anything. It's just still, this is what it says in the book. But I wanted to clarify it a little bit, what they actually do, how to interpret them. So this will be a little bit, so the alpha diversity, it's the mean species biodiversity within the sites or habitats of the local scale. So what I mean is that within a dietary group, for example, we want to see what is the diversity within that group. And I will give some childish example because I know that then you'll memorize it. So if you have a forest, a river, or a desert, if you want to measure the diversity, the alpha diversity of these three environments, it could be dietary groups. We know that the forest, there is 9 different species, so they are more rich, and that they're less in the river and even much less in the desert. So in the gut microbiota, normally higher and alpha diversity is associated with health. Normally, we need to pay attention on that, because with a high alpha diversity, it indicates that more bacteria cohabitate together, they have different functions and reduce chances of pathogen colonization. On the other scenario, if you have a low alpha diversity, it indicates there is less, quote mark, good bacteria. Just less rich or less diverse, which means that there is most likely there is less beneficial metabolites, and that is easier to colonize by pathogens. So this is one of the principles that we want to think about. So if you go back to the example that I have with the seawater and freshwater example, we could see that I'm not sure if this was significant, but this was just from an example, a random example that I showed. And we saw that in freshwater, it was richer or not or higher alpha diversity than in the seawater. But then what is the beta diversity? So, sorry, this is, I want to go for still on the alpha diversity. I wanted to give an example. So if you see here, this is an example with different diets, and the first one was a control diet. You see that there is, in the very end, you see the average is much lower diverse. Yeah, I think we could say diverse, or most likely there was a difference in the alpha diversity just by looking at different individual relative abundances. And from the very last group to the first one, something might have changed. So when we go to the alpha diversity, we see that even though they have the same, or like the control diet had a much wider variable results related, and then we saw that in this index observed ASVs. ASVs is the unit of sequencing. You see that there is a huge change, but not so nothing is significant. However, if you go to the Shannon index, that is a metric, mathematical calculation of the alpha diversity, you saw that there is much difference from the control to the first, this IM group. So if you see, go back to the example, it's because there is much within this group and with this group, they are much different between them. The same with another pill no evenness. Why do I put here the evenness? And then you'll see a lot of people, or a lot of times you only see this Shannon index, because Shannon index takes in consideration two factors, the evenness and the richness, which is this one. So sometimes when people just put the Shannon index without further information, we don't know if they are different because they are richer or just because they are more even. In another way, more similar between them. So it's really important sometimes to break this Shannon index in some other factors, indices. And I have some examples here. There's also Simpson index. So there is several. The most important to pay attention is not just alpha diversity. When they say alpha diversity increase, you need to be critical and think, what is this, which index was increased or which index was decreased? Because they mean completely different things. Another diversity that I want to stress about is the beta diversity. So in this case is the chance, the change, the presence or absence in species composition between two distinct sites or communities. So the other one was within a specific diet. Here is between two diets or more. But in this case, again, another childish example, but I'm sure that you will keep it in memory. So here, the beta diversity was 5, is because even though they have pretty similar alpha diversities, but when you compare the two groups, they have 5 groups that they were unique to the both farms. They both have cows, horses, and pigs, but what they have unique, or what is in absence in one on the other, that it matters as a beta diversity. So in the gut microbiota, a higher beta diversity reflects the presence or the absence of some rare species in the assemblage. So in theory, a higher beta diversity indicates more taxonomic differences, while a low beta diversity indicates more taxonomic similarity. So as an example of some indexes, there is a lot of different indexes, but they are mostly mathematically related. These we have the Jacquard distance, then there is other ones, but in the way, they are always the same data, is a different mathematical approach, and sometimes they can be a mirror of the others or either a vertical or horizontal mirror, and different mathematical approach. But normally it's just different mathematical approaches basically. It's okay to use the one with explains with a PCO1 with a higher explanation, or the one that shows the better what you want to prove. But yeah, there is more details about it, but there is no need. So exactly, because here this is what the way that we visualize it. It's by the distance to centroid, for example, in this case, mathematical distance to centroid. So sometimes it's difficult to visualize what is better, but this is the way that we could actually present results. So I would like just to give an example that we always need to be critical when people say high alpha diversity, which means more species, and it's better, and the low alpha diversity is bad. We always need to be critical, because in an extreme and oversimplified example, by modulating the microbiota of the different diets, one alpha diversity can be really high, but they can have a lot of pathogens, and they have a rich bacterial community, but they are not beneficially necessarily a good thing. Or they have a lower alpha diversity, but they could be very full, effective, really effective bacteria that they can perform a lot of functions and fulfill all the fish requirements. So again, we need to know the taxonomy, who is there, and what is their function. So yeah, for the first class, for the first part, I would like to thank your attention, and I'm able to answer some questions. And I also was thinking to give a little break before we go for the other class. But yes, feel free to ask any question.
Well, I guess you have some partners here that understand very much what you are talking right now, Sergio. Dr. Edison is here in the audience. Dr. Samira from Spain is also here. They might have some question for you.
Mm.
Well, Sergio, I'm working with aquaponic systems. Okay,
Yeah.
Laura. After me, I want to give you the word, Laura. I'm working with aquaponic system, and the
Mm.
aquaponics they also have the microbiota. So
Yeah.
how come the aquaponic system microbiota work with the microbiota that the fish has? Is it influencing or changing it? Do you have any experience with that?
So if it all matters first if the fish drinks water or not, and with the intestinal microbiota might be more dependent of if the fish drinks or not. But definitely it will change the skin microbiota and the gills microbiota. I'm
Mhm.
not I never well I'm starting how to work on the skin microbiota. I'm analyzing some data from an experiment. Definitely there is a correlation between the environment, the skin microbiota, because that they can activate the immune response in a different way. And I mean, they are exposed to the environment. So if but these bacteria that in the water, they might be related with the conditions that you have your culture. So depends which plants you have or which environment you're proposing, the temperature as well, the oxygen level. So I think that as richer is the environment, more stable it can be in the long term. So I don't see a negative thing. At all. But I guess that in the long term is also helps the fish to be less vulnerable because the environment is more stable.
That's interesting. Do you have any work with
No,
microbiota in the root of plants?
no, no,
Not
no,
yet.
no. But I guess that they are more related with subnutrification and some other roles important for the plants, and that is something that we also see in the aquaculture, the microbiota of fish in aquaculture, because of there is a lot of um, small algae growing on the nets, and so I have no idea, but I would not be. I think it's extremely interesting, but I don't think that is a bad thing at all.
All right. Thank you,
Yeah.
Sergio. Laura has a question for you.
Hello. My
Hi.
question is related to the salinity.
Hmm.
You talk about salinity and microbiota,
Hmm.
but what it's about that fish that are smoltifying, like salmon. What
Yeah.
happened with the change in microbiota when they